Today we'll learn staining procedures. So, staining is a method that is employed to improve the contrast of samples, typically at the microscopic level. So, there are two words that are used interchangeably. One is stain and the other one is dye. So, stain is known as the colorizing agent used to study biological structure, and dye is a colorizing agent used for general purpose. So this is a very basic difference between these two words. Otherwise, these two words are used interchangeably in microbiology. So chemically, a dye or a stain is defined as an organic compound containing both chromophore and oxochrome linked to the benzene ring. So what is chromophore and what is oxochrome? So chromophore are the molecules that absorb light at a specific wavelength and subsequently emit color. That is, it is a chemical group that imparts color to the benzene, if we can simplify it. And what does oxochrome do? It imparts electrolytic dissociation or ionization properties to the chromogen, allowing it to form salts and bind to fibers and tissues. So this will become more clear here in the making of this stain. So here we can see that there is a benzene which is colorless. Then we have added chromophore and it makes chromogen. So this chromogen is not a stain now, it's a dye, we can say. Okay, so in order to become a stain, we need to add another electrolytic compound, that is oxochrome, which then makes a stain. In this case, it's trinitrohydroxybenzene or picric acid. So this compound, the final product, is a stain. So in order to become a stain, there should be two compounds, that is chromophore and oxochrome. Otherwise, we will call it a dye. So coming to the uses of the stain, what does the stain basically do? It reveals the shape and sizes of the microscopic organism, shows various internal and external structures. Staining produces some chemical reactions in the organism, leading to changes in their appearance, and it helps in identifying one microorganism from other that facilitates further classification. So these are the uses of stain. Now the stain or the dyes is classified in three categories. One is basic dyes, another one is acidic dyes, and the third one is neutral dyes. So basic dyes, they give the positive electrical charge and are cationic in nature. Binds to the negative charge molecules like proteins and nucleic acids. Since they are positive electric charge, they bind to the negative charge molecules. Example of these are methylene blue, crystal violet, basic fuchsine, safranine, and malachite green. So these are the general stain which we all have heard during our staining procedures in microbiology. Next is the acidic dyes. It gives a negative electrical charge and therefore are anionic in nature. Since it is negative electrical in charge, it binds to the positive charge molecules like carboxyl groups and hydroxyl groups. Examples are acid fuchsine, eosin red, and rose bengal. Third category is neutral dyes. These are the complex salt of diacid with a dye base. Example of this are eosinate of methylene blue. Now the types of staining method that we know that are, first is the simple staining, which is very commonly used, and the second is the differential staining. So in simple staining, it uses single stain like methylene blue or crystal violet or safranine. So what is the purpose of simple staining? It is just used for visualization purpose. Visualization as in morphological features like size, shapes and arrangements. There is one cons of simple staining, that is it cannot do structural visualization. So to overcome this cons, differential staining was developed. So differential staining, what it does, it uses more than one stain. Both the dyes react differently with various parts of bacterial cell, allowing proper differentiation. That is, for separation of bacteria into groups is the function for separation of bacteria into groups and for visualization of the structures. So It can be divided into two categories broadly. One for is group separation. In group separation, again it's of two types, gram staining and another one is acid fast staining. And the second category is structural visualization, where we will learn capsule staining or negative stain, spore stain or scaffold or spore stain, and flagellar stain. So in this video, we'll learn about gram staining and acid fast staining and the staining related to structural visualization we will study in our next video. So compound that is very important in this staining procedure is mordant. So what is a mordant? It's a substance that forms an insoluble compound with a stain and serves to fix the color to the bacterial cell. That is, it increases the interaction between the cell and the dye so that the cell is stained more strongly. So this is the function of the mordant. We must have heard about gram iodine in gram staining procedure, right? So there are two types of applying conditions of this mordant. First condition, that mordant is applied first to the bacterial smear, followed by the addition of the staining solution. And the second condition, mordant is added to the staining solution and the complete preparation is applied to the bacteria at once. So these are the two conditions using mordant. Examples of mordant are iodine, gram iodine, tannic acid, salts of aluminium, iron, zinc, copper, chromium and tin. So these are the colors of different stains. First is the crystal violet, which is somewhat in purple in color. Saffron is pink in color. Methylene blue is bluish in color. Carbon fuchsin is red. Malachite green is bluish green. Acid fuchsin is red again. Eosin gives two types of colors in different areas of the cell. In the cytoplasm, it gives reddish color, and in the nuclei, it gives purple color. Indian ink has carbon black color, whereas nigrosin has black color. Eosin red gives orange red color, eosin yellow gives yellow color. Iodine here is again dark brown or dark orange in color. Congo red is reddish orange. Basic fuchsin comes in powder form and it is dark green in color. Whereas bromophenol blue gives different types of colors in different solution. That is, in acidic solution it gives yellow color. In basic solution it gives blue, whereas in neutral solution it gives green. So coming to the gram staining. This is the first differential staining used for group separation. It was first developed by Danish physician Hans Christian Gram in 1884. And this staining method was developed in Berlin. It's the most widely employed staining method in bacteriology. It is a type of differential staining procedure that divides the bacteria into four groups: gram positive, gram negative, gram variable, and gram non-reactive. So gram positive and gram negative, we all must have heard. And the other two categories are gram variable and gram non-reactive. So the reagents that are used for gram staining includes primary stain, that is a crystal violet, mordant, that's a gram iodine, decolorizing agent, that is 95% ethanol or acetone, and a counter stain is used, that is saffron. So the gram staining procedure. In the step 1 we have a bacterial smear on the slide. We dry it and then it is heat fixed on the flame. Step 2, the smear is stained with primary stain, that is crystal violet, and it is allowed to react for 1 minute. Then step 3, stain is washed off with distilled water. After that, gram iodine is applied on the smear and allowed to stand for 30 seconds. It is used as a mordant. Gram iodine is used as a mordant that increases the interaction between the cell wall and the stain. So this is the function of the mordant which we studied earlier. So that is why the gram stain is used. Then the slide is decolorized with 95% ethanol until all the color is removed from the slide. After that, in step 6, counter-stain saffronin was added and allowed to stay for 1 minute. In step 7, we wash off the bacterial smear of the slide with water and then the slide is blotted dry and examined under microscope. So observation under microscope. Gram-positive bacteria will retain the CV, that is crystal violet, and appear purple in color, somewhat like this. Gram-negative bacteria will retain safranin and appear pink in color, like shown in this picture. Gram-variable bacteria will retain the stain unevenly and appear as a mixture of color, like in this picture, like shown in this picture, purple and pink. So, two types of color we can see in this gram-variable. Gram non-reactive, they retain the color very poorly, or sometimes they do not even retain color. So they appear like transparent in color. So there's a theory behind the gram non-reactivity, that is the cultures that are too old, that is more than 48 hours of culture, if we stain it, then they will give this gram non-reactivity. So therefore it is recommended that we use 12 hours culture to 24 hours cultures maximum for this staining procedure. So what is the mechanism behind this gram staining? So the mechanism depends on the structure and composition of the bacterial cell. So there are two types of theories present. First is lipid composition and the second one is peptidoglycan composition. So first, coming to the lipid composition. In gram-positive bacteria, what does it have? It have a very lower lipid content than gram-negative bacteria. So that's why it dehydrates when treated with alcohol. And after dehydration, what happens? Pore sizes decreases, resulting in the reduced permeability, because of which the CVI complex, that is crystal violet and iodine complex, cannot be extracted out from the cell wall. So therefore, it appears purple violet in color. Whereas in gram-negative bacteria, they have a higher lipid content because of the presence of outer membrane. The alcohol treatment, what it does, it extracts the lipids. So the pore sizes increases, permeability is therefore enhanced, and the CVI complex is completely extracted out. Therefore, what they do, they retain the safranin and appear pink in color. So this is the first theory that we know. So the second theory is based on the peptidoglycan composition. So in gram-positive bacteria, they have a very thick peptidoglycan. This peptidoglycan is extensively cross-linked, and because of this cross-linked, pore sizes are very small, resulting in less permeability again. Therefore, the CVI complex is trapped and they appear purple violet in color. Whereas gram-negative bacteria, they have a very thin peptidoglycan as compared to the gram-positive. Therefore, they are less extensively cross-linked. Pore size remains sufficiently large, permeability is high, and they cannot retain CVI complex. It gets washed out. Therefore, they retain safranin and appear pink in color. So this will become more clear when we see the structure. So coming to the bacterial cell wall composition. Here we can see gram-positive bacterial cell wall, gram-negative bacterial cell wall. So here we can see that gram-positive bacterial cell wall has a thick peptidoglycan followed by periplasmic space underneath, then cytoplasmic membrane or plasma membrane. So this is the structure we can see. It's extensively cross-linked peptidoglycan. Therefore, the crystal violet is retained strongly. Whereas in gram-negative bacteria, there is an outer membrane present. Underneath, there is a peptidoglycan layer which is very thin as compared to the gram-positive bacteria. Underneath there is a periplasmic space followed by the plasma membrane. So here in the structure we can see the peptidoglycan is less extensively cross-linked, which upon dehydration, what it does, it extracts out the CVI complex. Therefore, it's retain the safranin color and appear pink in color. So this is the mechanism of the gram staining which we need to understand. Now coming to the second method of the group separation, that is acid-fast stain or Ziehl-Neelsen stain. So this acid-fast stain was first developed by Franz Ziehl and Frederick Nielsen. Franz Ziehl was the first to employ carbolic acid as a mordant, and Frederick Nielsen maintained Ziehl mordant, but he replaced the primary stain with the basic fuchsin. However, this scientist, which is Paul Ehrlich, he was the first to employ this complete preparation as the acid-fast stain on the mycobacteria in 1882. So therefore, this was completely developed by Paul Ehrlich, we can say. So these are the scientists who developed the acid-fast stain. First is the Franz Seele, first to employ carbolic acid as a model. Then it's the Frederick Carl Adolf Nielsen, replaced the primary stain with basic fuchsin. And then is the Paul Ehrlich, who is the first one to use Ziehl-Neelsen acid-fast stain on mycobacteria in 1882. So by this we can know that the acid-fast stain was developed before gram staining, right? This is in 1882 and the gram staining was in 1884. So this stain method was developed in Germany. It's the most widely employed staining method for mycobacteria. And in the early to mid 1890s, this technique was referred to as Ziehl-Neelsen method. It is the type of differential staining procedure that divides the bacteria into two groups. One is acid-fast bacteria and the other one is non-acid-fast bacteria. So the reagents that are used in this acid-fast stain are primary stain, decolorizing agent and a counter stain. For primary stain, carbylfuchsin is used. For decolorizing agent, acid alcohol is used. That is the combination of 95% ethanol plus 3% HCl. And a counter stain, methylene blue is used. So coming to the procedure of the acid-fast staining. So first the bacterial smear is prepared on the slide. Then it is air dried. Carbylfuchsin is added to the smear. In step 3, the slide is heated for 3 to 5 minutes so that the carbylfuchsin starts to steam. So apply more stain when the evaporation starts. So why this heating is done? So heating enhances the penetration of the primary stain into the waxy cell wall of the bacteria. This mycobacteria has a different cell wall structure than that of gram-positive and gram-negative bacteria. It does not take the color readily, so heating is required for this to enter into the waxy cell wall of this bacteria, right? In step 4, slide is washed with distilled water and decolorized with acid alcohol. Again, it is washed with distilled water. Smear is then counterstained with methylene blue for 1 minute and then washed with distilled water. Then the slide is air dried and observed under microscope. So the observation under microscope, what will say is that the acid-fast bacteria will retain the carbylfuchsin and appear red in color, somewhat like this. And the non-acid-fast bacteria will retain the methylene blue and appear blue in color as shown in this picture. So coming to the mechanism of acid-fast staining. So what makes Ziehl-Neelsen stain special for mycobacteria? So the mechanism depends on the composition of the cell wall. The cell wall structure is shown here. So some bacterial cells are surrounded by a covering composed of fatty acid or waxy substance which makes it non-permeable. Here if you see in this picture, we can see there is an outer membrane which is a characteristic property of a gram-negative bacteria. Then we have a outer membrane already has some portion of lipids. Then after this we have mycolic acid, again lipid. Then after this we have arabinogalactone, and after that we have peptidoglycan, and then we reach the cell membrane. So we can see it's a thick layer of lipids is present which makes it waxy. So therefore the mycobacterial cell wall are rich in lipids due to the presence of this mycolic acid. As I said, these organisms are not readily stained, but once stain is introduced inside the cell wall, it becomes very difficult to decolorize it even with a drastic decolorizing agent. They are called acid-fast because the stained bacteria are resistant to decolorization with acid alcohol. Like they studied in gram-positive and negative, decolorization, the most of the stain gets washed out, but here it is acid-fast. They are called acid-fast because the stain is retained so strongly that it cannot be removed even with decolorizing agent. So that is why they are called acid-fast. Example of this group are mycobacterial species and nocardia. So in this video, we have learned about two of the methods from differential staining for group separation. One is gram staining and one is acid-fast staining or Ziehl-Neelsen staining. So the other staining procedures related to structural visualization we'll study in the next video. Thanks for watching.